Fertilization is the fusion of haploid male and female gametes, that is egg or ovum, and sperm or spermatozoon to form the diploid zygote. Fertilization is the central event in sexual reproduction for continuing one's own species. The process of fertilization involves signaling between both spermatozoa and oocyte. Eggs. Eggs are large, symmetrical and non-motile cells. Human eggs are arrested in metaphase of the second meiotic division and complete meiosis only upon fertilization. Their surface is covered by microvilli. Eggs are surrounded by an extracellular layer, the zona pellucida, which is a glycoprotein coat composed of 3 glycoproteins ZPGP 1, 2, 3. All 3 of the glycoproteins contain O and N linked oligosaccharides. The zona pellucida is a species specific barrier to the sperms. For example, the human sperms are incapable of fertilizing intact hamster eggs, but experiments have shown that they can fertilize hamster eggs stripped of their zona pellucida. This concept is used clinically to assess the fertilizing capacity of sperms. Sperms. Sperms are small, asymmetrical and motile cells. They have 3 components: tail or principal piece. The tail contains the flagellar apparatus, which is composed of 9+2 microtubules and accessory structures. The sliding of the microtubule is powered by the dynein protein present in dynein arm and ATPase. Midpiece. It lies at the proximal portion of the tail and contains a sheath of mitochondria which produce the ATP necessary for beating of the tail. Head. It contains the spermatic haploid nucleus. Overlaying the head is a membrane bound vesicle, the acrosome. Sperm do not possess any organelles associated with protein synthesis like Golgi, rough endoplasmic reticulum or lysosomes. The sperm plasma membrane is also highly differentiated and contains proteins localized in distinct regions. One of these is termed as pH 30 or fertilin and is localized in the equatorial region of the sperm and is involved in sperm egg plasma membrane fusion. Acrosome. The acrosome is a lysosomal like compartment derived from the Golgi. It has low pH and contains soluble hydrolases. In cross section through the head of a sperm, 4 membranes are seen traversing from the plasma membranes to the nuclear membrane. During the acrosome reaction, fusion of the outer acrosomal membrane with the plasma membrane releases the contents of the acrosome and exposes the inner acrosomal membrane as the functional outer boundary of the sperm head. Depending on the site of fertilization, there are 2 types of fertilization. External fertilization. When fertilization occurs outside the body of maternal parent or outside the oviducts of the female, it is known as external fertilization. Majority of marine animals shed their sperm and ova into the surrounding water where fertilization occurs. It is seen in fishes, amphibians and echinoderms. Due to great hazards in open waters, astronomical numbers of eggs and sperms are spawned and deposited very close to each other for successful fertilization. Internal fertilization. The male deposits the sperms inside the oviducts or genital tract of the female, where fusion takes place. This mode of fertilization takes place in ovoviviparous animals like lizards, snakes, scorpion, certain fishes, etc., and in viviparous animals like insects, most reptiles, birds and egg-laying mammals. Even in this mode, millions of sperms are released so that some of them can cross all the barriers and reach the ovum. In humans, about 350 million sperms are discharged by the male, out of which only few can reach the fallopian tube and only one succeeds in fertilizing the ovum. The process of fertilization in animals is completed in 5 steps. Movement of the sperm towards the egg or meeting of gametes. During either of the fertilization type just discussed, the initial step is the physical encounter of the spermatozoon and the ovum by random swimming movements. They collide as a matter of pure chance. In individuals with external fertilization, as in sea urchin and frog, the meeting of sperm and egg is not a simple matter. Many marine organisms release their gametes into the environment that may be as small as a tide pool or as large as an ocean. Hence, large numbers of eggs and sperms are released outside so that they have a favorable chance of meeting with each other. Moreover, this environment is shared with other species that may shed their sex cells at the same time. These marine animals face 2 problems: the meeting of sperm and egg in such a dilute concentration and species specific activation. Attach themselves to the egg, but only one sperm can ultimately fertilize the egg, while the others are inhibited from entering. Hence, one of the hallmarks of fertilization is that it is species specific, that is, sperm from one species cannot or usually do not fertilize eggs of another species. After successful fertilization, the fertilization envelope is formed. Capacitation and contact or barrier penetration. In mammals, as sperms are released from the testis, they acquire motility when they pass through the epidermis, believed to be due to chemical bonding of fertilizin and antifertilizin. The spermatozoa are seen to agglutinate or adhere to the surface of egg and each other. The substances causing this agglutination of spermatozoa were described by F. R. Lilly in 1919. Lilly reported that eggs produce a glycoprotein high molecular weight substance termed fertilizin, which combines with an acid protein low molecular weight substance antifertilizin present on the surface of spermatozoa. This fertilizin antifertilizin reaction is highly specific and disperses the cell or corona radiata, the layer surrounding the egg. Below this layer is the zona pellucida that contains zona pellucida glycoproteins. Experiments showed that zona pellucida glycoproteins from unfertilized eggs contain a receptor that is modified after fertilization. 3 glycoproteins ZPGP1, ZPGP2 and ZPGP3 are found, of which only ZPGP3 receptor could prevent sperm binding to eggs. When sperms are ejaculated, they have oligosaccharides bound to the galactosyl transferase. During capacitation, these coating glycoproteins are removed, allowing the galactosyl transferase to bind to other carbohydrates such as those attached to ZPGP3. Penetration of sperm into ovum or acrosome reaction. Activation of sperm swimming movements requires stimulation of the propulsion machinery of the sperm. Sperms are activated in a series of steps. First, chemotaxis. The contact with diffusible molecules contained in egg jelly initiates chemotaxis. Chemotaxis is the process by which a migrating cell moves towards a higher concentration of a diffusible or substrate bound guidance molecules. The small peptide acts as a chemoattractant for sea urchin sperm. When receptors on the surface of the sperm bind a signal transduction cascade, involving cyclic GMP, it triggers faster sperm swimming. Second, acrosome reaction. When the sperm comes in contact with egg jelly, the acrosome undergoes radical changes resulting in acrosome reaction. This triggers a rise in internal calcium concentration inside the sperm head. The acrosomal membrane and the sperm plasma membrane rupture at the point of contact and joins around the margin of rupture, exposing the lysins to the surface of the egg membrane. The lysins have the ability to dissolve the egg membrane that allows the entry of the sperm into the egg cytoplasm. The central part of the acrosome elongates and becomes transformed into an acrosomal tubule or filaments. Usually, only the head and the middle piece of the sperm enter the egg, while the tail is left outside. In sea urchins, the sperm head binds directly to the egg outer surface, skewering the egg, and this triggers the acrosome reaction. In response to signals transduced by receptors on the surface of the sperm that bind to components of egg jelly, actin polymerizes from a pool of globular actin to form the acrosomal process. The acrosomal vehicle fuses with the plasma membrane, releasing enzymes from the tip of the sperm that aid digestion of egg jelly. At the same time, bindin is deposited on the surface of the acrosome reacted sperm. The acrosomal contents are released and there is a balanced sodium influx and proton efflux, causing an increase in pH. The increased pH triggers the dissociation of the prophylactin complex and the released actin monomers polymerize to form a filament called the acrosomal process. The depolarization causes voltage sensitive calcium channels to open in the egg and endoplasmic reticulum. Lysins from acrosomal vesicle digest the jelly coat and vitelline membrane. Calcium also activates a sodium proton pump which pumps proton out of the cell, increasing the intracellular pH. The increase in intracellular calcium causes water to enter the cell, increasing the hydrostatic pressure. This aids in the extension of the acrosomal process. The acrosomal process penetrates the egg coatings to allow fusion of the sperm and egg plasma membranes. The sperm head now has access to the cytoplasm. The calcium ion moves in a wave across the cell. This results in fusion of cortical vesicles with the egg membrane, releasing their contents into the space surrounding the egg called the pre-vitelline space. This raises the vitelline membrane and inactivates binding receptors on the vitelline membrane. Thus, the additional sperms are released from the vitelline membrane and cannot bind. The sperm head now enters the cytoplasm where it forms a male pronucleus. In humans, the thick zona pellucida keep sperm from binding close to the egg plasma membrane and triggers the acrosome reaction. The outer plasma membrane of the acrosome fuses at multiple sites with the plasma membrane and the contents of the acrosome are released. Two of the important components are acrosin and serine protease and N-acetyl glucosaminidase. Acrosin bores a hole in the zona pellucida so that the sperm can reach the egg itself. N-acetyl glucosaminidase hydrolyzes the O-linked oligosaccharides and ZPGP3. After sperm-egg fusion, the egg mounts the cortical reaction to prevent polyspermy. Activation of the ovum or cortical reaction. In all eggs residing just under the plasma membrane, there are membrane bound vesicles known as cortical granules. When a single sperm penetrates the egg, the cortical granules adjacent to the site are triggered to fuse with the plasma membrane, exocytosing their contents like hydrolase into the perivitelline space. The cortical reaction is propagated over the surface of the egg by a wave of calcium ion. The cortical granule degrades O-linked oligosaccharides on ZPGP3 and renders the zona pellucida incapable of binding additional sperm, thus preventing polyspermy. Activation of the egg also includes the initiation of development of the new zygote. Protein synthesis and other metabolic processes are upregulated to help the developing embryo. The zona pellucida, formed by protective secretions from the oocyte and follicle cells, surround the oocyte at its earliest stage. This glycoprotein layer supports communication between oocyte and follicle cells during oogenesis, protects oocyte, eggs and embryos during development, and regulates interactions between ovulated eggs and free swimming sperm during and following fertilization. The mature cumulus oocyte complex is made up of secondary oocyte, which is arrested at metaphase 2 following extrusion of first polar body PB. Cumulus oocytes possesses highly specialized cytoplasmic projections that pierce through the zona pellucida and form gap junctions with the oocyte and help in cell communication and signaling. In most mammalian species, cumulus cells surround the oocyte at the time of fertilization. The mature egg is generally in a hibernating condition with very low rates of metabolism and inactive nucleus. The penetration of the sperm triggers the egg into activity and brings about cortical changes and the development of fertilization membrane. In some echinoderm eggs, some fine granules are visible in the ooplasm after the entry of the sperm head. Egg activation blocks entry by additional sperm and stimulates the final meiotic division, including the onset of embryonic development. The vitelline membrane starts lifting up itself from the point of sperm entry and a perivitelline space is formed between it and the egg surface. The cortical granules from the egg cortex release their contents into the perivitelline space. These contents attach themselves to the inner surface of the vitelline membrane, forming what is known as a fertilization membrane. The development of the fertilization membrane prevents the entry of other sperms into the egg. The metabolic rates increase, allowing entry of water and other particles. The rate of protein synthesis goes up as these are needed for further cell divisions. At this stage, the pronucleus of the egg at the metaphase second division of meiotic II division becomes active and completes its second division, releasing the second polar body. Fusion of male and female pronuclei. The male pronucleus fuses with the egg pronucleus, regenerating twice n chromosomes. Microtubules and microtubule motor cytoplasmic dynein nucleated by the sperm centrosome serves as guide for the movement of the female pronucleus. In this process, there is fusion of male and female pronuclei. Initially, the two nuclei remain close together, and at the point of contact, the nuclear membranes disappear and the chromosomes come to lie on the equator. Finally, the nuclear fusion is completed and it becomes a zygote nucleus. The egg is said to have been fertilized and it becomes a zygote. Mitosis then occurs and is now ready to undergo cleavage to develop into the embryo. The process of fertilization has much significance, such as it injects a male haploid nucleus into the egg cytoplasm. It activates the egg and begins cytoplasmic reactions that permit development of embryo. It helps in combining genes derived from the two parents in the offspring. It restores the diploid state and introduces genetic variation. This intermingling of parental and maternal hereditary characters in the offspring is known as amphimixis. It contributes in the creation of new organisms. Fertilization is a cell-to-cell recognition process that occurs between two distinct cells, a small asymmetric and motile sperm cell, and a large and non-motile egg. It encompasses a series of different steps which have to be performed in a well orchestrated way to create a new individual. The neck of the sperm contains mitochondria and centriole that generates the microtubules of the flagellum and provides energy for flagellar motion of the sperm to approach the egg. The acrosome of the sperm contains enzymes and digests extracellular coats surrounding the egg called the vitelline envelope or zona pellucida. Eggs secrete diffusible molecules that attract and activate the sperm. In mammals, sperm must be capacitated in the female reproductive tract before they are capable of fertilizing the egg. Fusion between sperm and egg is species specific and is mediated by protein molecules like bindin, fertilin and integrins that allow the membranes to fuse. Fertilization thus helps in the combining of genes derived from two parents and creation of a new organism. The process of fertilization has been studied mostly in marine invertebrates such as sea urchin, balanoglossids, etc., and in amphibians and mammals among vertebrates.